Bismillahirrahmanirrahim. Assalamu alaikum. Let me introduce at first. I am Professor Dr Mahamuda Begum, Professor and Head, Department of Anatomy at the Women's Medical College, Mokbajar, Dhaka. I hope Allah subhanahu wa ta'ala save you and your family. Dear students, now we are going in a war against a unseen enemy. Inshallah, we win if we maintain our personal precaution. Alhamdulillah, we complete nicely a long journey of first and second week of development. It is my belief you understand all these events of these weeks. Let us start a new journey of third week of development today. The clear knowledge of every events of third weeks is essential. So the presentation of third weeks of development is delivered into two parts, because every events of these weeks, those are associated with each other. That means by chance, if one event is development is absent, so next events is not developed properly. Before we start the third week of development, the slide focus on the end result of second week of development. This is the formation of the bilaminar germ discs, and also this important amnion and chorion. Amnion includes amnioblast and somatopleuric layer of extraembryonic mesoderm, and chorion includes topoblast and somatopleuric layer of extraembryonic mesoderms. Now the third weeks of development. First we focus on what are the events occurred in this week. First is the most important, primitive stick. Primitive stick first appear, then cell gradually invaginated, so the process invagination. And third importance is the formation process, the gastrulation. By this the three germ layer is formed. And during this time, when the endoderm is established, the some cell of the endoderm is thickened and form a plate, the precursor plate. These are the inductor. All it is important for the formation of notochord. And these are the membrane, most cephalic region of the embryo, the embryonic disc. This is the membrane, the buccopharyngeal membrane, and this membrane is covered. The region is known as the stomodium. And when the stomodium is ruptured, the mouth cavity is formed. Next is the formation of the notochord, and also for the development of the topoblast. And on the caudal region, there is another membrane known as the cloacal membrane, which is covered the proctodium. And when this proctodium is ruptured, the next occurs the anus. So these are the figure. This figure showing this is the amniotic cavity and the layer epiblast, and gradually they are formed in the mesoderm and endoderm. The cavity is the yolk sac cavity, amniotic cavity, and these are the thickened cell of the endoderm, procaudal plate, and these are membrane, buccopharyngeal membrane. There is another is the cloacal membrane. This is the cloacal membrane. And these are the buccopharyngeal membrane. Their formation. This is the stomodium. This is the proctodium. In the cephalic region, here is formed mouth cavity, and therefore anus. These are the figure of the first second week. Bilaminar giant bricks, two cavity, and this is the shape of the embryonic discs, slippery shape, and the cephalic region. Therefore, this is the expanded, and the caudal region, and this is a caudal region. The primitive stick is formed, and this stick runs towards the cephalic region. Now, primitive sticks. First, what is this? It is the thickened band of ectodermal cell. Must focus this. This is the emphasize this. The what type of cell? Ectodermal cell. Ectodermal cell means epiblast. So, thickened band of epiblast cell or ectodermal cell. Region, the caudal region of the embryo in the midline. So, caudal region, thickened band of the ectodermal cell. And nature of this cell is pluripotent. Pluripotent means that the cell is transformed in any type of the tissue or cells, and this visible on the dorsal surface on day 15. So timing, they are appearing visible side, dorsal surface of the embryonic discs, and nature of the cell pluripotence, and midline, thickened band, and also the caudal. So when the caudal it is appeared, now the embryonic disc is divided, which part is caudal part and which part is the cephalic part, and also the cephalic part is gradually expanded. This embryo is the 18 days embryo. Now the parts of the primitive sticks. Primitive sticks appeared in the caudal region, and then it goes towards the cephalic region. On the journey, the two side of this stick is elevated, and this known as the primitive group. Then gradually at that side it is nodulated in form. Nodular structure is formed, the primitive node, and below the node there is a pit known as the primitive pit. So the parts is the primitive group, primitive node, and primitive pit. Now, function of the primitive sticks. Primitive sticks is responsible for the formation of intraembryonic mesoderm, formation of the septum transversum, initiate the formation of notochord. It is not formation, it is initiate. Okay, the primitive stick initiate the formation of notochord, and also determine the future craniocaudal axis of the embryo. Not only the craniocaudal, it also determine the left and right halves of the embryo. So again, formation is function is it help in the formation of the intraembryonic mesoderm, septum transversum, initiate the formation of notochord, and also determine craniocaudal axis and right and left halves of the embryonic disc. Now fate of the primitive sticks. Fate of the primitive sticks. The primitive stick regress craniocaudally. It also important. It start first appear in the caudal region and goes to the cranial region. So process is progress caudal to cranial, but when it is regress craniocaudally, and completely disappear by the end of the 4th weeks. After 4th weeks, no actions of the primitive sticks, but so completely disappear by the end of 4th week. But it becomes an insignificant. If it is present the some region in the body, but it is insignificant. Like this, sacrococcygeal region of the embryo. So if the sacrococcygeal region of the embryo, the primitive sticks may be persist, but it become insignificant structure. So fate regress craniocaudally, completely disappear by the end of the 4th week, and if it is present, the sacrococcygeal region, but it becomes an insignificant structure. Now the clinical correlations. This region, sacrococcygeal region, in the sacrococcygeal region, the primitive sticks, if the cells of the primitive sticks persist, it forms a tumor. This is known as the teratoma, sacrococcygeal. Why it is called teratoma? Because we know that cell of the primitive sticks is pluripotent cell. So it transforms in the different type of the tissue. The teratoma is the tumor which they are present all derivatives of the 3 germ layer structures. So the clinical correlation of the primitive sticks, if it is persist after 4th week of development, so in the region of the sacrococcyx and form a teratoma, this is the tumor, sacrococcygeal teratoma. This is the sacrococcygeal teratoma. Gastrulation. Gastrulation process. Gastrulation is the process by which established all 3 germ layers. That means ectoderm, mesoderm and endoderm in the embryo. So first clearly understand what is gastrulation. So gastrulation is the process that established all 3 germ layers, ectoderm, mesoderm and endoderm in the embryo. So this figure, there is a mechanism of the formation or the steps of formation of the 3 germ layer. This figure focus on gastrulation, process of gastrulation, and this figure also showing how the cell transform from the primitive sticks toward the cephalic regions. So the 2 figures included in the process of gastrulation or the step of gastrulations. Steps of the gastrulations. Gastrulation begins with the formation of primitive sticks on surface of epiblast. So first step of the gastrulation is appearance of primitive sticks on the surface of epiblast. So gastrulation begins. It is also the viva questions. Which is first? Gastrulation or primitive stick? Primitive sticks, because gastrulation begins with the formation of primitive sticks on the surface of epiblast. First step is the primitive sticks should appear at first. This is the primitive sticks appear. Then the primitive sticks, on the region of the primitive sticks, cells of the epiblast migrate towards the primitive. From the primitive sticks, the cells of the epiblast migrates. First is the migrates toward the primitive sticks. Then upon arrival at that regions, in this region, the cells become shape is this is the epiblast cell is the tall columnar, but now the cell is flat shaped and detached from the epiblast and now sleep beneath epiblast, that is epiblast, and this inward movement is known as invagination. So second step is the invagination. During invagination, what happens? Cells of the epiblast first migrate towards the primitive sticks. When this cell arrival at the epiblast, they change their shape, flag shape, and gradually detached from the epiblast and sleep beneath the epiblast cell. That is, this inward cell movement is known as the invagination. So first step, the primitivistic appearance, the formation of the primitivistics. Second events, invagination. The second step is the invagination. And after that, once cells have invaginated, first this cell displaced, this flag-shaped cell displaced the hypoblast cell and creating a new cell known as the embryonic endoderm. So first this invaginated cell, this cell first displaced the endodermal, that is hypoblast cell, and creating the new layer of the cell known as the new layer of the cell, the new layer of the cell known as the endoderm, embryonic endoderm. So invaginated cell first displaced hypoblast cell and formed the new, creating a new cell, layer of the cell. It is known as endoderm. This is known as endoderm. Next, other cell come to lie between epiblast and hypoblast. This is the hypoblast, that is endoderm. The other cell come to lie between the epiblast and newly creating endoderm cell and form mesoderm. So the first detached cell displace the hypoblast, and next the cell in between epiblast and endoderm form another new cells. It is known as mesoderm. So first create the endoderm, second create the mesoderm. All cells comes from epiblast. All cell first invaginate epiblast cell. They shape their cell is the flag shape, and first displace the hypoblast and form embryonic endoderm. Then the cell which are present in between the hypoblast and epiblast, that is endoderm and epiblast, this cell is converted into embryonic mesoderm. Then the cell remaining in the epiblast, this cell is transformed into or known as the ectoderm. Thus, the epiblast, through the process of gastrulations, is the source of all three germ layers. This is the explanatory questions. Why epiblast is responsible for the formation of three germ layers? So through the process of gastrulation, epiblast is responsible because first the cell invaginate from the epiblast cell and goes to the hypoblast, and this cell is displaced by a new cell layer known as the endoderm. Second, in between the cell hypoblast and endoderm, this cell transform mesoderm, and remaining cell in the epiblast is transformed into the ectoderm. So epiblast is the source of three germ layers. So cells in this layer will give rise to all of the tissue and organs in the embryo. So this is the end of this gastrulation and complete the first part of 3rd weeks of developments. Thank you all.